actually finalizing with coordination in plants, and we shall finalize it with some questions, which I will expect each one of you to answer depending on what you would have understood. Moles. We shall be going to coordination in animals. Please use this chat for your friends. We are using it for us saying good morning, good morning. You are spamming the chat box. Previously, if you still remember, we had ended with this thing called auxin, and it was the experiment to show that auxins are responsible for growth. And we're using coleoptiles. Coleoptiles simply means plant seedlings, those plants which are just growing from the seed. Those newly, when you cut off the upper tip coleoptile, growth stops. It's just like assuming you are growing beans, and then you cut off the upper tip, that bean plant is going to stop growing. It's going to stop growing. And the reason is simple. But if it is an old tree with already different branches, when you cut off the bouquet, but in the seedlings, it stops. So it's what we're going to look at. And I think I had told you with this experiment that we use, this experiment had been done by people like Darwin, as well as Went. There's another person is asking, how can I get the past explanations, because I've just joined. Such questions, we have already told you to go to Google Classroom. The notes are there. The notes are there. They're already shared. And when you cut off this upper part, the upper tip of this seedling, and then you place it on this agar block. I think I remember I used an example of a sponge. Imagine pouring water in a sponge. Okay, the sponge will be able to keep that water. Or you can picture a sponge from a mattress. When you pour water into it, it retains that water. Okay, now imagine that agar block. Imagine this agar block, or you can call it the sponge. Now placing it on another coleoptile whose tip had been cut off here. In this case, whose tip had been cut off. When you place it there, whose tip had been cut off, the growth continues. You can see that here there was no growth. No growth. Growth means to grow up, to grow, to continue growing. So no growth with that. And when they used an agar block without which had not been attached to the tip, again no growth here. But when they used an agar block that had been placed on a tip and then placed on this growing coleoptile, it grew. And when they put it offset sideways, it grew bending towards the other side. When they placed it also sideways, it grew going the other side, meaning that auxins diffused from the agar block going downward and made these cells to grow very fast, that made it to bend towards the other side. Nachimuli, you have a question?
Yes, sir.
When I ask you to unmute, you unmute very fast and you ask, yes, what is your question?
Sir, just saying, I'm requesting, pardon. Please.
Pardon?
I've just joined the lesson. So I'm requesting you to pardon for me.
We have also just started, actually. We have just started. I was just actually reviewing what we looked at last time. Were you here last time?
Yes, sir.
Remember we had talked about this, so I'm reinforcing it before we can do some questions to finalize with this topic.
Thank you, sir.
Okay. So, and then let me see some questions here. We are doing coordination in plants. I don't know why people keep on asking a topic. Should I read the notes if I already have them? No, you don't need to write notes. These notes are already uploaded in Google Classroom. You don't need to rewrite notes, okay? So I was still explaining to you this experiment that had been done a long time ago to show that actually the shooty tip has that make these plants, or the seedlings, to grow. And the first thing to be done was to cut off that upper tip, the uppermost part of the seedling. When they cut it off, from there, growth stopped. The seedling stopped growing and died. But when they cut off that tip and they placed it on the agar block has capacity. It is some kind of material, just like a sponge, which has capacity to suck these auxins or plant hormones from the tip into it. And that's why you can see that in this first picture, the agar block was colorless, but the tip was purple, was pink. Look at this picture well, everyone. You can see that the tip is pink. Just a moment, just a moment. So I was still telling you that you can see that this was purple, okay, and this one is colorless. Okay. You can see that it is colorless. Now, after some time, you can see that these hormones which were in this tip, they moved out of the tip and they went into this block. And this is in the second picture here. Okay, you can see that the hormones, which we are calling auxins, moved from the tip into the block. The block was placed, you can see they diffused down and they made it to elongate upward. Seeing that. And in some seedlings, it was when they did that, the auxins still diffused from the block and they made that part to develop more than this and side L. So they moved the side L faster and they grew. And these cells were not dividing very fast. So it bent towards the left. So they said, maybe it was a mistake. Let us try placing it the other side of K. Again, when they placed the agar block halfway on the side K, the auxins diffused down, made those cells to divide very fast, and then they made that part to grow and elongate more than this side L, and it bent towards the right. Okay, and I think it is now very clear. So I hope you guys have understood that. So in summary, what happened was that auxin, you can see that this is auxin in the tip of seedling. They were there, they were cut off. So late grown seedling. It was a person who presented this work in 1928. 1928. This is when this experiment removed the tips of oat seedlings. Oat, there are some kind of cereals which are grown in Europe. Okay, just like here we grow sorghum, millet, like that. So in Europe, they grow oats. So when to remove the tips of oat seedlings and put them on agar. Agar is the other block which I was telling you. An inert gelatinous substance. Gelatinous means like it looks like Vaseline or mucus. Mucus eminira. That's how it looks like. So, and you can see that they were placing it on this agar block. Now, what happened is that the oxygens diffused from the tip and entered the agar block. You can see that oxygens diffused, they entered the agar block. Now, blocks of agar were then placed off center. This is the center. They were placed off center, not in the middle, but just on the side. Off center on the ends of other oat seedlings, from which the tips had been considered. They diffused downward and made these cells to grow more often, and then it elongated more than this side, and that's why it bent towards the left, the right side. So the seedlings bent away from the side on which the agar block was placed. You see some. Now, you guys, I told you we are using this chat box to ask the questions concerning the topic. And I'm seeing people are just conversing. Those who are saying that you are not seeing, it is your network. People are seeing. Which class is this? This is Form 3. Which link did you click in? Because all links have classes. Which topic? You know, if you have been in biology, we have been handling coordination. So I don't know what is wrong with some people. Now, there is a graph which is normally examined. I've seen this graph in some, is it mock question? For some of you who know mock questions, mock papers, when you resume school, you will be able to do mock papers. Now, they can sometimes give you data, or they can give you a graph which has already been plotted. Having growth, or what you can call elongation, that is to elongate oxygens increasing towards the other side. Okay. Now, see, normally the concentrations are measured in what we call parts per million. This is ppm. This is parts per million. This is a unit, just like kilogram, measuring centimeters, like that. They are just units, but these units measure very small quantities. So you can see that as oxygens keep on increasing, okay, I was still telling you that from, let's say, 0 point something, as they keep on increasing, so this is an indicator that the higher the concentration of oxygens, the lower the rate of elongation or growth in the roots. Okay. And that's why I was saying that with auxins, they have an effect. Remember the other experiment of geotropism. The experiment of geotropism was showing you that okay, if this was the seedling, assuming this is the seed and this is the root part of it. If this is the shoot, now this is the root. Remember how geotropism makes the root to bend. It is that the root is horizontal like this. Auxins are going, so you're going to have just like a bottle. I gave an example of a bottle. So I gave an example of a bottle containing water. That water is, when you have water in a bottle like this, and let's say the water is halfway, okay? When you make this bottle to slip like this, when you make the bottle to slip like this, the water is going to settle down like that. It will not remain as in this bottle, but it's going to be slipping down. So that is the example which I use for auxins. So auxins are going to settle down in the root like this. Now in roots, what actually happens is that they are going to, the higher the concentration, when auxins flow down and of the root like this, they stop, or the concentration of the auxins is going to increase here. And so they are going to reduce growth of these cells here on the lower side of the root. So what is going to happen is that this root, the upper part is going to grow faster. This upper part without auxins is going to begin growing faster and faster and faster and faster and faster, more than this side is going to begin bending downward and bending downward and bending downward. Okay. So it's going to keep on bending downward and bending downward like that, okay? And that's why in this graph, as oxygen concentrations increase, growth or elongation stops. So these cells were not growing because they were being stopped by oxygen concentration, but the upper cells were growing, and that's what made the root to bend, to grow up more than this lower side, and that's what made the root to bend downward to respond to geotropism. But in the shoot, if I can go back, but in the shoot, the opposite happens. As oxygen concentrations increase, growth increases. So if you still remember, I'm assuming this is the seedling, okay? And this was the shoot, like that. Let me put a root there, a small root. So in the shoot, instead, auxins are going to be more here. Now, when you are having more auxins here, they promote in the stem, in this graph 2, this is graph 1, they promote growth of cells and divisions of cells. So this side where auxins are going to divide more often and divide and elongate more often and elongate more often, but the upper part is not elongating. So that is what is going to make the shoot to bend upward like that. So it's going to keep on growing upward and upward and upward like that, because auxins here promote cell elongation, but here they inhibit or they stop cell elongation. So the upper part grows more often, and that's why the root bends downward. So you can see here that it says that when the concentration of auxins increases, growth in the shoot also increases to a maximum here. As concentration of auxins keep on increasing from, let's say, 1, 2, let's say 10, 30, 40, like that, parts per million, I'm assuming. So in the stem, as concentration increases here, growth actually increases a certain point, okay? Beyond which further increase in oxygen's concentration inhibits growth in shoot. So up to that point, so then it is after bending has occurred and it's now looking up, and then it stops such that the root can, the shoot can continue growing upward in the direction of the sun. So which further now growth response in the root decreases with increase in concentration, oxygen concentration. And I think that is very, very clear. Let me see what people are writing. Okay. So the screen, it was just my network for moving out, but I told you to use this platform to ask questions. So is there any question concerning this before I can go to the next slide? Do you have any question concerning this before I can go to the next slide? But Dangira is saying, what is tropism? My God. I don't know whether maybe you have just joined. I talked about this when we are beginning. Tropism just refers to a growth response towards a unidirectional stimulus. The stimulus is anything which can cause or which can evoke a response in an organism. That is the stimulus. Okay. Maso says, I've understood very well. Thank you, teacher. Is it a must that there must be a graph? Yes. Paper 1 of biology, number 31. This is the setting of the paper. The paper has 30 objective questions which are compulsory. I'm talking about paper 1 biology. That is 553/1. That is its paper code. 30 objective questions, and so section A has 30 marks. Then section B has 40 marks. Number 31 normally has 20 marks, which is normally a graphical number. Number 31 with the 20 marks. Then number 32 normally is a structured question with 10 marks, and number 33 is a structured question with 10 marks. Then also section C, you choose 2 questions, and of which each question has 15 marks. That is the setting. So number 31, most cases it is a graphical question, or it could be, but most cases it's a graph number. Teacher, when the growth in the shoot reaches a maximum, does growth stop? Yes. Here, the growth that we're talking about is those elongations of the cells. So after the root has bent upward like that, okay, it stops because concentration has reached maximum. It stops, and then the tip continues. The tip begins producing more oxygen such that, and in this case, because it's already looking up, since it's already looking up, what happens is that we just continue growing up and up and up. Now more is produced from the tip. Then Hakim is saying, teacher, explain more about the graph. I don't know what you want me to explain more about. Prossy, I've understood. Thanks. I've just joined. What are we learning about? We are learning about coordination in plants. Teacher, when the growth in the shoot reaches a maximum, okay, how do oxygens control phototropism? Now, I talked about this under phototropism, Mike. I don't know whether you missed that class. Just to know, in summary, Mike, how oxygens control phototropism. Oxygens don't like light, or they fear light. So if you're having a shoot here and you having light here, I'm assuming this is sunlight, what happens is that oxygens which were evenly distributed, they run away from this lighted side where light is coming from, and they run to the dark side. When they run to the dark side, they, in the shoot, they make this side to grow more often and 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 more often. And then the shoot bends towards light. Okay, that is what I can say. Oh, fuck. I don't know what's wrong with this network. Okay, yeah, I thought I had lost the network. So I hope you have understood that very clearly. Continue from slide show, from current slide, yes. So I'm often, I think you have understood that. Then let me see some more questions. Okay. I've got that, sir. Okay. You guys are nagging such questions are irrelevant. Oh, someone's saying that such questions are irrelevant. So, okay, let us continue to the next slide. I hope everyone has understood. Now, oxygens have very many importances. And some of the importances can be, okay, let me first pick some questions here. I am having Hakim. If I ask you to unmute, you unmute very fast and ask.
Excuse me, teacher.
Yeah.
I have not understood the explanation of the graph.
You mean the whole of it?
Yes.
Okay, I'm going to summarize for you. And then there's one who is called PC. PC, can you ask? When I ask you to unmute, you unmute very fast.
Excuse me, teacher.
Yeah.
Teacher, what if you take off the tip of the plant?
When you take off the tip of the plant, growth stops immediately. And that plant dies. Growth can never resume if you cut it off. Then Randolph, is it Randolph? When I ask you to unmute, you unmute very fast and ask. Yes.
Teacher, could you please repeat for me to explain the graph, because I've just joined.
Repeat what? Which one? Come again. Not go to where you want me to repeat from. And then, okay. Then Faith.
Sir, I have not understood the curves of the graph.
Not understood the curves of the graph.
Yes.
Okay. I'm going to, let me comment about this graph once more. Let me see here. Kellen is saying, teacher, does the tip of the plant have a scientific name? Now, Kellen, it's just like, look at, Kellen, look at your fingers, okay? And just let's say choose the, let's say the first finger. Now, the first finger has that tip which you use to touch objects, okay? There's nothing special about it. It's just a tip. It's nothing like it has a scientific name. It's just a tip, okay, of a plant. Just like if you have a head, when all plants, by the way, let me make this clear to Helen, to Kellen. When a plant is growing, it doesn't begin growing leaves immediately. It doesn't begin growing branches immediately. It first elongates from the soil. Let me, sorry for that. Let me first go back here. Let me get my pen. Yeah. When if you just get a seedling, you just use a bean seed. When it is growing, it first comes out like that. It doesn't come out as leaves. And even when this one is coming out, normally they first come out just simple, something simple. simple structures. Sometimes they could be having small leaves, but it's just a simple thing. That is the tip. It doesn't have a special name, okay, unless you want to call it in another language. Marian said, patient, this is not your, it's biology. Don't disorganize us, please. Okay, for sure, sir. Just move on and let them learn to keep time. Okay, that's what Mercy is saying. Hey guys, let's keep time. Teacher, could you please send your WhatsApp number after this one? I have something urgent to share with you, personal. And then it's not some type of bad. 223. If he keeps repeating for you who has just joined you. Okay, yeah, that is true. Some people come very late and then they keep on asking things which we have done in the previous lessons. And remember, that is not going to make us move at a good pace. You shall be dragging so much. And I hope I've answered most of your questions. Okay, we can go to the next slide. Now, importance of auxins. Remember, I told you auxins, you can call them plant hormones. Now, hormones, I think even in primary school, you talk about hormones. They're chemical substances, okay, which are normally secreted at by organisms in different parts of their body. So they're produced at a given part, and then they cause change in a given part of the body. So the auxins, call them plant hormones. Of course, there are very many types, okay, of plant hormones, but we look at auxins more often. One of the important is they cause apical dominance. Now, apical dominance comes from the word apex. If you have done flowering plants in your O level, sorry, in your senior 1, senior 2, you know that apex is that upper tip of something. That is the apex, okay, if it were a leaf. Now, normally, apical dominance, the dominance comes from the word to dominate. To dominate means to rule over the other. Now, what happens is that auxins, they are going to rule over, if they are here, they are going to cause apical dominance. They rule over the other parts of the plant. Whereby, in most cases, if some of you have what we call those flowers which you normally grow in your gardens, normally they will have just a few leaves when they are just growing. They will just have a few leaves, okay? But the moment you cut off the tip or you trim the upper parts of the plant, you find that this plant that has been trimmed, to trim means to cut off the upper part, it now grows other big branches. It becomes bushy. It becomes so bushy, meaning that when you cut off the tip, you have removed what we call apical dominance. It's no longer dominating. So these other branches can now begin growing freely, and the plant just becomes bushy. It is not going to grow tall as you wanted it. So in most cases, that's why people keep on trimming those leaves or those small plants that they normally have in their compound. They keep on trimming them so that they become thick, they become bushy, and they don't overgrow tall to become big trees. Hope that is clear. Then leads to parthenocarpy. Now, some plants actually can develop fruits even without them being fertilized. An example in a case is the pineapple, okay? That is what we call parthenocarpy. These auxins again drive that process. Then also causes tropisms. Remember I said tropisms are plant growth responses in their growth response in response to a given stimulus, the stimulus can be light, in that case it is phototropism. If it is gravity, in that case it is geotropism. If it is water, in that case it is hydrotropism, and so on and so forth. Then it causes rooting of the stem cuttings. Now, some people go on and cut off small short stems, and then they pour auxins on them, especially those people who grow flowers. I don't know whether you've been to Rosebud. There's a company along Entebbe Road called Rosebud along Namulanda. They grow flowers and they sell them abroad. So if you can visit such a company, they sometimes have cuttings for, let's say, roses, if you want to propagate them. And when you power their auxins, they can grow some small roots, and then you can plant, and then they can grow faster. That's what we are talking about here. Now, let me see some of your comments. What is parthenocarpy? I've just talked about it. Then which topic? Because I'm confused, or you can help me with the WhatsApp number. What's the next topic? The next topic, we are looking at coordination. We are not yet through with it. We are just concluding coordination in plants. So after coordination in plants, we shall go to coordination in animals. Then Kamigisha, you guys who are talking about some other things, please respect the lesson and only ask questions concerning the lesson. When are the next lessons goes? She explain more about apical dominance. You are breaking. And then how can we get notes? The notes are there in Google Classroom as the class Benard. Download a classroom app. Okay, please use this to chat relevant questions. Okay, to ask relevant questions. More about apical dominance. Not go to apical dominance. Okay, let me explain more about apical dominance. This is what I said. Apical means the upper part of a given plant. Dominance means to rule over. Now, the auxins, because they are normally produced in the upper part, so they have a tendency of ruling over the whole plant such that it does not grow branches anyhow, because in the initial growth stages of a plant, it wants to first elongate so that it can go up high and high above where there is much sunshine. And then after it has elongated so high and auxins that are produced at the tip can no longer reach down the plant very well, then those plants can now begin growing branches. They can now begin growing different branches. Okay, and this is important in the elongation of a given plant. But if you cut off the upper tip when the plant is still growing, this plant is not going to grow tall. It's not going to elongate. It will just begin growing branches there and then until become thick and bushy. Okay, so when you talk of causes apical dominance, it means that this apical dominance has a significance of making this plant to grow tall and tall, so that it can reach up to trap more sunshine, because plants, that's what they want. They want to move up as high as possible so that they can trap sunlight energy to make food. So apical dominance is very much important in growth, in elongation of the plants as they are elongating to compete for sunlight. But when you cut off the tip, this plant is not going to grow tall. It's just going to become short and then becomes bushy with more branches before it can elongate to compete for the sunshine well. I hope you guys have understood that apical dominance. I said, please use this thing to only ask relevant questions. And then parthenocarpy, I said that some plants can develop fruits without undergoing fertilization. You know that for any plant to produce fruits, let's say guavas, there has to first be fertilization, whereby pollen grains move from one flower and then they go and fertilize the stigma of another flower, and then the fruit develops. But some do not wait for pollen grains to come and then they are fertilized. Some can actually develop into fruits. That's without fertilization. That's what we are calling parthenocarpy. Okay, growing fruits without fertilization. It is important. Imagine if pineapples had to wait for pollen grains to form fruits, which sometimes they never come. That's why sometimes some seasons you may find that some mangoes don't produce mangoes. And some seasons they produce, meaning that maybe some seasons they don't receive pollen grains. I think you can see how important it is. Okay. Please, I said I don't know, admin, if you can, I'm going to block, if you keep on chatting things which are not related to the question, I'm going to block the chat box and none of you will have capacity to send anything anymore. Okay. I'm going to block this. If you're just chatting things which are not part of the lesson, I'll just block it. And then there's this question which I got from a UNEB question, a UNEB bank, which I want us to look at concerning coordination in plants. It says that the figure below shows the effect of unidirectional light on the growth of plant shoots. Study the figure and answer the questions that follow. This is a UNEB question, remember, that was brought some time back. Experiment at the beginning, this is how it was looking like. This was a box. You can see it has a key. 3 says coleoptile with a tip covered with aluminium foil. Now this was a coleoptile. That is the tip of the coleoptile. Coleoptile means seedling, which was covered with a piece of aluminium foil. Then another coleoptile, the tip had been cut off. And then another coleoptile, it was left intact, and then light was left to go through this dark box, as you can see. Okay, you can see maybe this one was poorly drawn a little bit. This one was poorly. It's like they made it to bend a little bit, but it didn't bend. It was just a. Let me redraw it here. It's just straight. This thing. Okay, let me use a black marker. Let me first eraser. Let me first erase this, and then I use a black marker. So it's supposed to be like this. Supposed to be straight. So it is straight. It is straight like that. So don't say that this one is bending. This one is not bending. It's not bending. It is straight. Okay. So this is at the beginning. This is how they were, but after 5 days, check. This is what is happening to the second. If this is 1, 2, 3, this is what happening. What is happening to the second tip? No growth is happening. The first tip, it is bending towards the other side. This third tip is just elongating. It's just elongating and elongating upward. Okay. And then the question is, explain why each coleoptile appears as shown after 5 days. And I want us to begin with a coleoptile eye. Explain why it is appearing like this. Initially it was like this, but then why is it appearing like that? Okay, let us go to, let me get some people here to answer. Let me put the chat box here. I'll begin with Philomena Bakita. When I unblock you, you answer very fast. Yes, Philomena.
Yes, please, sir.
Yeah, you raised your hand. You want to answer?
Yeah. Coleoptile Roman number 1 appears bent because of the presence of oxygens that light energy moved towards it and caused it to bend towards light so as to cover the oxygens.
So as to?
So it bent towards light so as to cover the oxygens.
So that they cover the oxygens?
Yes.
What do you mean by covering the oxygens?
It bent towards the light because due to the presence of auxins that do not need light.
So when light shines on that side, where do the oxygens move to?
They moved upwards.
Do they move upward, or like what makes them to move? Okay.
I don't know how to say it, but when light comes on that, because auxins are on the tip of the plant, so when light comes on it, it causes it to bend. Because earlier you said auxins don't like light.
That is very true.
Yes.
So because they don't like light, if they are here, they will run away from it and go to this side where it is the dark side, this side.
Yes.
So they run away from this side and go to this side, making this side to bend. Okay.
Yes.
Okay. Thank you, Bakita. Thank you. Okay. Then there is Faith.
May I think when the light was exposed to the seedling, Roman 1, but the auxins, they just moved on the opposite side.
Thank you. That is very good.
Of the seedling. So they caused it to grow one side, like that one side grew faster than the other, leading to the bending towards light.
Thank you very much, Faith. That is very good. And because of time, now the poll that I'm choosing, I want you to talk about Roman 2. Brian Michael.
There is no growth in Coleoptera 2 because the tip was cut off, and it is then that produces the oxygens.
Okay. So there was no oxygens, and therefore growth stopped. Is it? Brian? Okay, thank you for that. Then how about William? William G.
I'm going to answer the third coleoptera.
Yes, please.
The third coleoptera was covered with aluminium foil, which blocked the light from reaching the tip of the plant. So the auxins remained in even distribution in the coleoptera, so it kept growing straight.
Thank you, William. I like that word, even distribution. I like the word that you brought in, that auxins were evenly distributed, so they caused uniform growth of the tip, and so it continued growing upwards. Thank you, William. Okay, I think you've had William. That was a very good submission. And then I think they are done now. They are done. Maybe let us go to the next slide and finalize. PC saying, yeah, the auxins were not present. And then absence of oxygen stopped growth. Yes, that is true, Philomena. Then we can end with this question. Now, I got this question from Jebu. Jebu mock is, in fact, is a ginger joint examination board mock. It's a very good mock body, which this question was set in 2019. And I want us to end with this question. It says that in an experiment setup, a seedling was placed horizontally, as shown below. Study the diagram and use it to answer the questions that follow. And you can see A, we're having, then B. Question says, name the parts labeled A and B. Let me choose some people here. We are having William G. William G has raised the hand first. William G, tell us number 8.
It's by mistake. I wasn't raising
by mistake.
Yes.
Okay. And then which person is here? Judith Prossy.
I think part A is the plumule.
Mhm.
I think part A is the plumule.
What about B?
I'm not sure of part B.
You are not sure? Why wouldn't you be sure? Okay, thank you, Prossy.
Maybe it's the root.
Maybe it's the root. Yeah, for some of you, you can always type the answers in the chat box. I will be reading them. Then Priska. Priska.
Part A, teacher, I'm here. Part A
Yes.
is the root, then B is the shoot.
B is the shoot and A is the root, sure. Hey, Priska. Okay, let us choose another person. Hakim.
Part A is the shoot and B is the root.
Okay, yes, that is actually correct. And you can see already that normally the shoot has some kind of leafy structures that are trying to grow up. Okay, and the root normally will not have those leafy structures. So it is very clear that A is part, okay? And we can go to the next part B. Part B. Oh, let me first see here. Hey, Nathan, let me first go back. Because, okay, saying Kengisha A is shoot, B is root. Root is a promoter had been removed, promote oxygens. Hayla says A is the root, B is the shoot. You guys. Logose, you are saying A is the root. Nathan Akanaho says A is the shoot, B is the root. Brania Joseph A shoot, B root. Nalun Kuma Olivia A shoot. Ja says A shoot, B root. Yes. And then Aisha Divine, they're all saying the same thing. Yes, that is correct. A will be the shoot part and B will be the root. Then B says if says that if the seedling is left for 3 days, describe the shape and position of A and B in terms of oxygen concentration. Describe the shape and the position. Okay. Remember, I told you to describe means to paint a picture of something in words. So, and you can see 7 marks. That means A, that is shape and position will be for A, we will have like 3.5 marks. Yeah, and then B, the shape and positioning will have again 3.5 marks. Let me choose some people. Now I need someone to talk about A first. A is the root cause I have observed in seedling germinating. They always bring those structures in the soil as they grow. Yeah, we are done with that now. We are now looking at part B. Who is there? Faith. Faith, you had unmuted yourself. Okay. Yes, Faith.
Sir,
Yes.
I think in A, the oxygens are gonna be distributed downwards. So they are going to cause it to grow this side bending upwards.
Okay.
Leading to the shoot. That's all.
Okay, thank you. So describing the shape, yes, it is bending upward position. If the thing is there for 3 days, describe the shape and position of A. So it's going to be bending upward. And then B, how about John? Yes, John. John, can you say something about B? Okay, nothing. Yes, Tekino. Tekino?
It will go downwards due to the what of geotropism. Gravity will pull B downwards.
Okay. To pull B downward, and so it will be bending, facing downward. So gravity, how does it pull it? Does it just pull it? Then you have to, so the axons flow downward, causing these cells here to divide more often, and then they make this B to bend downward. Okay. We are left with about 1 minute. Of course, we are just getting ideas. Then let us look at, name the tropic response shown in A. So what is the tropic response shown in A and B? Joshua has moved down. Yes, Wide. Wide. Okay, let me choose another person. What is the tropic response? Yes, Faith.
For me, I think that's geotropism because it's just in response towards light.
For A, is it negative or positive?
It's positive and B is negative.
Are you sure? Because positive means you are going towards the stimulus, and B, and negative means you're going away from the stimulus. Let me end with Judith Prossy. Yes, Judith. Yes, Judith. Okay, maybe the network. Brian says B is positive and negative. Teacher, thanks for the lesson. Gongo students love you. Thank you very much. Musinguzi Anthony, positive. B is positive. And then Bagay, thanks for the lesson.
Patrick, we need to end.
Okay. Okay. Thank you very much. The lesson has ended, and goodbye.